The human body does a lot. It walks and runs. It also does DNA replication and active transport. Nonetheless, the human body cannot do any of these functions without energy. We eat food to give our body the energy it needs. But the big question is, how does our body extract energy from the food we eat? Hi, my name is Pisaya Deina and I'm a high school junior. Review these terms before continuing the video. Cellular respiration is a process in which our bodies convert food into energy. It is essential to life. Our bodies decompose the foods we eat into simpler substances. For example, when we eat a starchy substance like rice, our bodies decompose it into glucose, a simple sugar. Then the glucose is oxidized in the presence of oxygen to produce carbon dioxide, water, and ATP. In addition to carbohydrates, proteins and lipids can also be used for cellular respiration, but for this video, I will demonstrate the process using glucose. The first stage of cell respiration is glycolysis, which literally means splitting of sugars. In the cell cytoplasm, glucose, a 6-carbon sugar, is decomposed into 2 3-carbon sugars. These 3-carbon sugars are oxidized, so they become 2 pyruvates. In addition to the pyruvates, 2 ATP and 2 NADH are produced. The ATP are produced through a process called substrate-level phosphorylation, in which an enzyme reacts PEP and ADP to create ATP. 2 NADH are produced because as glucose bonds are broken, energy or electrons are released and bond to NAD+. Glycolysis does not need oxygen to function, but the next 2 stages will only happen when oxygen is present. Stage 2, the Krebs cycle, is named after the scientist who explained it, Hans Krebs. In this stage, the 2 pyruvates from glycolysis are transported to the mitochondrion. Both molecules lose a carboxyl group, and the carboxyl groups leave the cell as carbon dioxide. Because of this loss, the pyruvates become 2 carbon compounds. The 2 carbon compounds are oxidized, and the substance becomes acetate. The electrons from the oxidized compounds reduce NAD+. And the acetate reacts with coenzyme A, forming acetyl-CoA. The acetyl-CoA enters the Krebs cycle where 8 enzymes in the mitochondrial matrix completely catabolize the compound, releasing additional carbon dioxide and energy. The energy is stored in NADH and FADH2. The cycle begins with 2 carbons bonding to the enzyme oxaloacetate. Then, this enzyme becomes citrate, but after a series of steps returns to oxaloacetate. Since oxaloacetate is regenerated, the Krebs cycle is called a cycle. In total, 6 carbon dioxide, 2 ATP, 8 NADH, and 2 FADH2 are produced during stage 2. The electrons bonded to NADH and FADH2 will be used in the 3rd stage, oxidative phosphorylation. Oxidative phosphorylation contains 2 parts, the electron transport chain and chemiosmosis. It occurs in the mitochondrion. First, the electrons from NADH and FADH are passed on into the electron transport chain. The electrons lose energy as they are passed down the chain to molecular oxygen, the final electron acceptor. Half of the molecular oxygen combines with the 2 hydrogen ions to form a water molecule. For every glucose molecule, 4 waters are produced. Next is chemiosmosis. The energy released from the electrons forces the hydrogen ions from the matrix into the intermembrane space. This creates a difference in hydrogen ion concentration. And in order to balance the difference, the hydrogen ions flow back into the matrix through ATP synthase. The flow of hydrogen ions through ATP synthase powers it to combine ADP and an inorganic phosphate, the reactant needed to produce ATP. Chemiosmosis produces a maximum of 34 ATP when it happens perfectly. Overall, cellular respiration produces 38 ATP. After watching this video, try to formulate an answer to the big question. Thanks for watching!